genomic library, complete digestion at all relevant restriction sites, produces a large number of short DNA clones. Genes containing 2 or more restriction sites may be cloned in 2 or more pieces. Mechanical shearing produces longer DNA fragments, and these are not uniform, require enzymatic modification before fragment can be inserted into a cloning vector. Partial digestion cut at a less frequent restriction site and limit the amount and the time the enzyme is active, result in population of large overlapping fragments. Fragment can be size selected by agarose electrophoresis. Fragments have sticky ends and can be cloned directly. Partial digestion with SAU3A result in library of overlapping DNA fragments of various sizes. Partial digestion of DNA by restriction enzyme. A series of overlapping fragments, each with identical 5 GATC sticky ends. Resulting fragment may be inserted into BAM-H1 site, a plasmid cloning vector. The plasmid hybrid site can be cleaved by SAU3A but not by BAM-H1. DNA fragment from SAU digest hybrid site can be cleaved by both SAU3A and BAM-H1. Screening library. Plasmid vector containing digested genomic DNA are transformed into E. coli. Screening a genomic library. Plasmid vector containing digested genomic DNA are transformed into E. coli colonies that grow then are replicated onto a membrane. Bacteria are lysed and DNA is denatured. Membrane-bound DNA is next probed with complementary DNA. Complementary DNA in the probe is composed of DNA sequence. You are looking for homologous sequence presumably also found in library. Unbound probe DNA is washed off the filter. Hybridization of probe DNA is detected by exposure to X-ray film or by chemiluminescence. Pattern is noted from exposure pattern of clone on X-ray film. Select a clone that test positive and isolate for further analysis. Transform E. coli with genomic library. Velvet surface, sterilized, pressed on the plate. Plate on selective growth medium colonies grow. Replicate plate colonies onto new selective medium. Filter removed from the culture dish. DNA denatured and bound to the filter. Probe DNA hybridized to DNA on the filter. Label probe solution added to the filter in heat sealable bag. Wash filter free of the unbound probe. Detect hybridization by auto radiography for radioactivity labeled probes or by chemiluminescence detection for non-radioactive labeled probe. Chromosome library. Screening can be reduced if target genes can be localized to particular chromosome. Chromosome can be separated by flow cytometry. Condensed chromosomes are stained with fluorescent dye. Chromosomes separated based on the level of binding of the dye and are detected with a laser. Complementary DNA library is derived from the mature messenger RNA, doesn't include introns. Complementary DNA may contain less information than the coding region. Reflect gene activity of a cell at the time messenger RNA are isolated. Messenger RNA degrade quickly after cell death and typically require immediate isolation. Creating complementary DNA library isolate messenger RNA, synthesize complementary DNA, clone complementary DNA. Creating complementary DNA library. 1. Isolate messenger RNA. Mature eukaryote messenger has a poly A tail at the 3 end. Messenger RNA is isolated by passing cell lysate over poly T column composed of oligo desoxythiomidylitic acid. Poly A tail stick to the oligo DTs and messenger RNAs are retained. All other molecules pass through the column. Creating a complementary DNA library. Step 2. Anneal a short oligo DT primer to the poly A tail. Primer extended by reverse transcriptase 5 to 3, creating a messenger RNA DNA hybrid. Messenger RNA is next degraded by RNAs, but leaving small RNA fragment intact to be used as primer. DNA polymerase synthesize new DNA 5 to 3 and remove the RNA primers. DNA ligase connect DNA fragments, result in double-stranded complementary DNA copy of messenger RNA. Poly A tail synthesis of complementary DNA degrade RNA fragment used as a primer for new DNA synthesis. DNA polymerase 1 synthesize new DNA strand in fragment and remove RNA primer. DNA fragment joined by DNA ligase. Double-stranded complementary DNA is formed. Creating complementary DNA library. Complementary DNA has blunt end, thus need to add restriction site linker or adapter to make them sticky. Use T4 DNA ligase and blunt end ligation to add restriction site linker. Digest the linker with the same restriction enzyme used to cleave the vector. Mix complementary DNA and cut the vector DNA in presence of DNA ligase. Transform into E. coli host for cloning. If complementary DNA has the same restriction site and the linker, complementary DNA will be cloned in places. Solution: use adapter with single-stranded overhang that match the restriction site on the vector. Cloning of the cDNA using BAM-H1 linker. Double-stranded complementary DNA, a T4 DNA ligase, cleavage of linker with BAM-H1, insertion into the vector cleaved with BAM-H1. Screening complementary DNA library. Complementary DNA library are most often used to detect gene for protein. If you know the DNA sequence for the protein coding gene you want to find, a homologous DNA probe can be used. If no homologous DNA sequence is available, complementary DNA can be probed with an antibody that recognizes the protein expression vector. Cloned complementary DNA is inserted between promoter and transcription terminator before it's transformed. Messenger RNA is transcribed from complementary DNA and translated. Colonies now expressing protein are transferred to membrane. Membrane is incubated with radioactive labeled antibody probe that recognize the protein. Non-radioactive chemiluminescent probe also are available. Colonies with bound antibodies leave a dark spot on X-ray film. Screening complementary DNA library with antibody probe, recombinant DNA plasmid, promoter, and translation start origin, clone complementary DNA sequence, transcription terminator sequence, transform E. coli with complementary DNA colonies in expression vector, transfer individual colonies to microtiter dish and grow store, plate on selective growth medium, colonies grow, transform individual colonies to microtiter dish, bring colonies to membrane filter and grow on medium, take filter off and lyse cells in situ. Protein product of complementary is bound to the filter. React protein on the filter, with radioactive antibody wash off the unbound antibody.